Today we're going to talk about cell transport. And so yesterday in lab, you demonstrated this with everything really, except for the temperature was just sort of showing how temperature affects diffusion. But when we talk about the cell, everything to do with transport falls back on the cell membrane. And so when we look at cell membrane, do you remember, I think I mentioned this term last week, but what is the primary compound of the cell membrane?
Lipids.
It's a type of lipid, yep.
Oh, phospholipid.
Phospholipid, right. So all of these they look like a balloon with two strings on it. These are phospholipids. And we say that the cell membrane is in what's called a phospholipid bilayer. So there's two layers. And the phospholipids themselves actually have a couple of important features. And I think what I'm actually going to do here real quick, I'm going to pull up some lecture slides from last week because there's a really nice, I don't have my textbook with me to show you, but there's a nice picture of phospholipids that I can show you. This guy. Okay, so here is a phospholipid. This is just from the lecture material from last week. So what we're looking at here, again, is kind of like a balloon with two strings on it. And the balloon part of that is a polar molecule and has phosphate. If I say it's polar, what does that mean to you? What did you say, Sharice? Yeah, polarity, polar molecules can attach to other polar molecules with hydrogen bonds. Good. What's an example of a polar molecule that we've talked about? Water, right. And so basically a polar molecule has two different charges, right? It's polar, like there's a positive end and a negative end. That's how water is. A battery, a magnet, for example. So this balloon has a polar head and then non-polar tails. And the tails are fatty acids. So we kind of talked about fatty acids last week a little bit. But I don't know if we've mentioned this much. Well, maybe I have. So our bodies and most living organisms are about 60 to 80% water. So there's water in our cells and there's water outside of our cells. And so that being said, and you can kind of see the diagram up here, but which direction would these polar heads, would they be facing the water or facing away from the water?
Facing the water.
Facing the water, right? So when we see this diagram here, I'm going to go back to our other slides. Cell transport, here's a picture of our cell membrane. So this is the outside of the cell and it's watery, and this is the inside of the cell and it's watery. So these phospholipid heads are facing the water. So we say that the polar head, another term for that is hydrophilic, and that essentially translates to water loving. So philic is loving. And then the non-polar tails are hydrophobic. So they're water fearing. Hydrophobic. So that's going to give us an idea of how things can cross the cell membrane. So most of the cell membrane is covered in phospholipids, but then we also have some of our other major organic molecules from last week in there as well. So we have, if you look at the model, these little sort of yellow sequin-looking structures are carbohydrates, and these carbohydrates stick out of the outside of the cell. And carbohydrates on a cell have a couple of functions. I'm just going to put carbs. Carbohydrates on a cell kind of act like a "hello, my name is" sticker. It's like a cell identification. On bacterial cells, carbohydrates can act like a sticky matrix to stick to other bacterial cells or stick to surfaces. And so they, again, kind of give a unique mosaic to the cell. lipids, so we have phospholipids, but then we also have cholesterol in our cell membranes. And remember that cholesterol is one of those weird lipids where it has these 4 rings attached to it. I think that was, I can't say anything else because a couple people haven't taken the test yet, so just kidding. Anyway, cholesterol helps. Does anyone remember what it does? I don't know if I told you. What's that, David?
Oh, nothing. I just said it's in eggs.
It's what? In eggs. Yeah, it is in eggs. In a cell membrane, cholesterol helps to stabilize and make it more rigid. So normally, remember last week we talked about saturated versus unsaturated fats? And which one of those was more fluid? Unsaturated, right. And unsaturated fats are more fluid because their fatty acid chains have kinks in the chain, so they're just fluid. Same with the phospholipids. So the cell membrane itself is always kind of moving around a little bit, so the cholesterol helps to stabilize it. And so that's important. And in terms of animals or animal cells, and humans, hold on, stable, I'm just going to write stable cell, which means it stabilizes the membrane. Our livers make about 85% of the cholesterol we need, but the rest we get from our diet. So, like egg yolks, for example, will give us that. And then finally, proteins in a cell membrane, if you can see here, they kind of will span the entire width of the cell membrane. And proteins are going to act like channels or like pores for different substances to move through. So they're kind of like little turnstiles or little tunnels through the cell membrane for different things to go in and out of. So when we look at some general features of the cell membrane before we talk about some of the examples from yesterday, we say that the cell membrane is selectively permeable, or I think on the lab yesterday I used the word semi-permeable. So what does it mean if something is permeable?
It can go in and out.
Yeah, it's penetrable, things can cross in and out. And if something is impermeable, it's not, right? Exactly. So like a good raincoat should be impermeable to water, right? So a cell membrane is actually semi-permeable or selectively permeable. And that means that it selects for what can come into and what can leave the cell. And that's going to be based on a couple properties. So really small molecules can cross the cell membrane. Non-polar substances can cross the cell membrane. So of all the things that we looked at yesterday, if we think about your dialysis tubing experiment, what was something that was really teeny tiny that was able to cross through that tubing? Glucose, yes, is small. Iodine? Iodine is even smaller, right? So that iodine is an ion, and that was, did that go, well, it started outside the bag. So how do you know that iodine diffused into your bag? Yeah, it turned the starch that blue color, right, which was in your cell. And so small molecules can do that, and then things like gases, like carbon dioxide, oxygen, those are diffusing in and out of your cells and in and out of your lungs as we speak. And then non-polar substances here, like glycerol, alcohol, those can also pass through. And so what I wanted to show you before we continue, I should have showed you this from the last slide, but the selective permeability of the cell membrane is because of the phospholipid bilayer. So we say that these phospholipids are self-orienting, which means if you had a handful of phospholipids and you threw them in a container, you would see this arrangement. So if I don't want to like spill this, but if you move these all around, they're eventually going to self-orient, and that's because these polar heads want to be by the water. I know there's no water up here, but just pretend. And then the non-polar tails want nothing to do with the water, so they're going to face each other. And so this orientation, again, allows for different things to come into and out of the cell. So for example, a polar molecule can't cross the cell membrane easily because there's nonpolar stuff in the middle. So something like water, which is always going in and out of cells, actually goes through one of those protein channels. And I want to pass this around, but this must be the non-leaky one. I don't think it's going to leak. So if you just, don't spin it upside down. But if you want to take a look at this super cool cell membrane. Okay, maybe there's a little oil on it. Let me just get it. It's not going to hurt you. It's just, now that I'm back here, I'll let you guys know. Yeah, I know. You can take a look at it first. So, here, enjoy.
I don't want it anymore.
It's really cool. I need some coffee. So, when we look then at, so yesterday on your lab, one of the terms I had you identify was diffusion, there was osmosis, active transport. And so when we look at diffusion, do you remember what diffusion means? Right, that was almost in unison. So basically substances move from a high concentration to a low concentration. Excuse me. So we say that in order for diffusion to happen, there has to be what's called a gradient, a concentration gradient. So does anyone know what the word gradient means?
Like a scale.
Yeah, scale, basically like a difference. And so, like for example, yesterday, you had more glucose inside your cell than you had outside, so that was a difference in concentration gradient. There's a temperature gradient when you go from in this room to inside of a freezer, right? There's a pressure gradient that exists in your blood, for example. So with what's called passive transport, passive means no additional energy required, and so things will move, what we say, along their gradient. So passive is no additional energy required. Everything has kinetic energy, so there's always, our molecules are always kind of bouncing around. But we'll see with active transport that substances move against their gradient, so like from low to high concentration, and that requires energy. So what is the main energy currency in a cell? ATP, right. So active transport requires ATP. So A for active and A for ATP required. Okay, so we're going to spend most of the time looking at passive transport and kind of relate it back to what we did yesterday. And before we do that, this slide here just kind of shows that of those, depending on the type of molecule, there's different ways that it will cross the membrane based on the structure. And so because the membrane has, again, this nonpolar hydrophobic kind of inside, substances that are charged or substances that are too big, substances that are polar, can't just get right through. They have to go through some kind of channel or pore, like water goes through a pore. Other molecules that are small, that are non-charged, like this thing, can just freely cross the cell membrane. So I think I talked last week about steroid hormones and how steroids are nonpolar and they can just waltz into and out of our cells, which is kind of a big deal. I know maybe you're not as excited as I am about that, but it's cool. So nonpolar molecules can just in and out, no problem. No regulation. So if we get into then kind of what we looked at yesterday. So how about I talk about this slide and then we can relate it. So diffusion is again, movement of molecules or substances from an area of high concentration to an area of low concentration. And there was the one thing I forgot to grab out of my house this morning was air freshener, and I was going to stand up here and I was going to spray it, and then you were going to raise your hand and tell me when you smelled it. But I don't have it. It's not here. So diffusion, if you just imagine, right, that I'm standing here with air freshener, if I were to spray it right here, and not in your face, but up, right, in the beginning, the people kind of right around here would probably start to smell it, right? Because it would be a high concentration. But there's a low concentration of whatever apple spice molecules out there. And so everything in nature wants to be in equilibrium. I'm sorry, Sharice, did you have a question?
Does perfume work or does it have to work?
It does. It would work. I'm just wondering if anyone has any like weird, not weird, it's not weird if you have a sensitivity, that's not what I meant.
Weirdo. No, nobody does.
Nobody does. Who would be, and please own it and be honest, but is anyone opposed to me like spraying one spritz of Sharice's perfume?
Do it.
Does anyone have like a, you know, some people are really sensitive, so.
Okay.
Are you?
No.
I feel like you would only be sensitive if it's sprayed like on you. But
Yeah,
if
I'm
you're
gonna
so
go like
far
this,
away,
like,
you know.
yeah, it'll
right.
probably just
Already
fall on
already
me.
smell my perfume, guys.
Thanks.
I always keep shit on.
Bath and Body Works. Every time I really
Thanks.
walk by people.
I will give it back. Okay, so what I'm gonna do, we're gonna basically demonstrate diffusion by this perfume. And so maybe I'll spray it over there and then it's not like right on top of someone. So right away, and then when you start to smell it, just raise your hand, okay? I'm just gonna do one. It's gonna smell really good in here. Oh, okay, maybe two. There's not much in there. Try not to walk under it. You don't have to be totally silent. It'll get there, right? But what we should, so right now there's a very high concentration of Rebel flower, whatever, of perfume right here. Why can't I say that word? Fleur? Whatever.
Oh, it's like French for.
Yeah, it's Rihanna, right? So, oh, I just smelled it, but it's right here.
I smell coffee.
Anyone smelling it yet?
I don't know.
Just walk by.
I smell.
I didn't want to spray
I smell
like
coffee.
Oh,
I smell something.
But anyway, there's a high concentration of aroma particles, like, there, it's right here now. Whew, it's good, it's good. I'm gonna open the door a little bit, so we don't all get, it does smell good.
It's the smell of flair.
So so basically the aroma particles are here-ish and but it's too high of a concentration. And so as I was saying before, Sharice generously gave us her perfume, that things in nature want to reach equilibrium. And so it's too high of a concentration over here, so that's why through kinetic energy, those particles are going to diffuse out into the room. And I didn't spray that much, so I don't know if it's gonna get there, but is anyone else starting to, and it's probably on me and I keep moving, so there's that. But that's the same thing we saw with our color dye experiment yesterday, right? Even that we tried to use temperature to demonstrate, anyone else getting any whiffs? No one wants to raise their hand? Okay. It was, yeah, it's fun. It smells good, it smells good. So we saw that with our color dye, and what you did yesterday with the color dye reminds me of like coloring, yeah, here it comes, coloring Easter eggs, right? Where you like drop the dye and it just sits there. So these pictures from your textbook show this little drop of dye, and there's a lot of, let's just call them purple molecules right there. And there's not a lot of purple molecules out here. So through diffusion and through just kinetic energy, which is different than ATP, that kinetic energy will allow those particles to diffuse throughout the container. So in here, what was it called again? Rebel flower, throughout the room. These poor fish and the food coloring dye, right? And then yeah, I don't know. Probably not. And then yesterday in your glass experiment. And so the other thing about that, even if it didn't quite work this way, and I think some of the groups saw the opposite reaction, but what was the beaker, hot, cold, or room temperature that showed the quickest rate of diffusion? Hot, right? In theory, that's what it was supposed to be, basically because what does temperature do to our kinetic energy? Yeah, it just agitates it, it speeds it up, right? So that just, diffusion is still happening, it's just allowed to happen faster because those molecules can just disperse more quickly. And then with the cold water, cold tends to slow down molecules. Which were some of the groups that saw it actually speed it up? Yeah, but you guys had ice, right?
Yeah.
And I think we talked like maybe as the ice melted, it like agitated some of those molecules. I don't know. So in the second group, did anyone see, in my afternoon crew, did anyone see that the cold was fastest? I thought someone did too, but
We did. I think our hot was fastest, and then room temperature was slowest,
was it?
and then cold was.
And our room temperature was a little colder than normal room
Yeah.
temperature in here yesterday. But regardless,
Still is.
diffusion is movement from an area of high concentration to an area of low concentration of particles until equilibrium is reached. Once equilibrium is reached, they're not really going to go have a net movement either way. So diffusion is the passive transport, high to low concentration, just in general. And then osmosis is a type of diffusion, but it's the diffusion of water across the semipermeable membrane. These are apples, and I will explain that in a second. It's kind of a bad picture.
Your cuts are apples like that.
It was a
It looks like you're putting
They're like
it was a peeler
yeah.
quarter slicer
Yeah,
thing.
gotcha.
So osmosis is the diffusion of water. And so sometimes, like yesterday, when you had the LODIA on the different slides, you had distilled water and you had tap water and salt water. Salt is big enough and it's charged enough that it's not going to cross the cell membrane, but water can. And so sodium can move by itself, but the salt wasn't going to move in that situation. So when water leaves or enters a cell, that's osmosis. But if there's a net movement of water and a lot comes in or out of a cell, that can disrupt normal cell function. So for example, when we had the LODIA in the salt water, I think it was, that's when all the chloroplasts kind of shriveled up, right? Did I say that right? Yes, because water out. And I'll explain that more in a minute. But one of the ideas too about osmotic pressure is that if you have a solution that is going to pull in water, so pull water into it, that means it has a lot of osmotic pressure. And so the greater the osmotic pressure, the greater the tendency to want to pull in water. And before I even talk about chloroplasts or talk about net movement, my example here, has anyone ever made a fruit pie, like an apple pie or rhubarb, whatever? So one of the instructions, you know, you get out your fancy apple peeler quarter slicer, you cut up your fruit, you throw it in a bowl, and then you add a whole load of sugar, some cinnamon, some flour, a bunch of dry stuff, right? And then you let it sit for 15-20 minutes and stir occasionally. I've made this 100 times, so. And in that time, you can prep your crust, you can go to the bathroom, whatever. So before you added the dry material, you just had your apples sitting there, right? Happy as a clam. Then you add all this dry stuff. Was there any water in that dry stuff? No, and if there was, maybe just a little. So now what you have, and then, so leave that thought there. In 15-20 minutes when you return, what do those, I tried to take a good picture, but it didn't really work. What do those apples look like? Are they still dried out? Is there a delicious, goopy, cinnamony, sugary mess around them? Yes, okay, you can, yeah. So what happened there is that, I'm gonna erase this here. So if we look at an apple, I'm a teacher and I'm just not that good at drawing an apple.
That's a really good apple.
Thanks. That is one of my best, actually.
Yeah.
So what do we have? Okay, obviously apples are a fruit. So what is one of the major organic molecules we might find?
Fructose.
Yeah, fructose, which is an example of a disaccharide, which is an example of a simple I heard someone say carbohydrate. Carbohydrate, right? I know, we're getting into detail here. So I'm just gonna write CH2O, we got carbohydrates, and we have some sugars. Okay, fine. but we have a lot of water too, right, because there's cells. Then you add all your sugars and all your flour and all your good stuff and let it sit there. And now all of a sudden we have a gradient, right, a concentration gradient. So we have more water in the apple, we have no water out here, right? So over time, this solution of your sugars and cinnamon and stuff has a high osmotic pressure and starts to pull the water out of the apple. So the water starts to leave the apple. It's not that the sugars go into it, it's that the water leaves the apple, and then that makes that kind of goopy mess. And then you put it in your shell and you bake it and it's delicious. But the point is, and maybe we can tie this into what happened yesterday, we have, if we consider the apple cells or cell, and then we have this solution with a lot of solute, with a lot of sugars and things outside, do you think this scenario that took water out of the cells is hypertonic or hypotonic or isotonic? Was this most like the distilled water, the tap water, or the salt water?
Salt water.
I'm hearing salt water. Anyone else want to think a different one? Do you need a nap? That's right. So it is like salt water because, so this is basically a hypertonic scenario because we have more solute, we've got these sugars and things on the outside than we do on the inside. And so water leaves the cell, water leaves the apple. I actually have a rhubarb pie recipe in my recipe book at home from a former A&P colleague of mine. And during the step where it says wait 15 minutes, she actually wrote osmosis at work. And I was like, yeah, A&P always, biology always. So this is osmosis, where you have water leaving or entering a cell because it's in a different concentration. So when we think about that, we can relate it back to what happened yesterday. And pause this for a second. Whenever we have a solution, I don't think I mentioned this before, but a solution always has two things. A solution has a solute, maybe we did talk about this like the first week, and a solvent, right? So what is, if we're talking salt water, what's the solvent? Water, right? Water is the universal solvent. So when we talk about hypertonic, hypotonic, isotonic, those terms refer to the amount of solute, so the stuff, in the solution outside of the cell. So when we look, so for example, yesterday the cell was the Elodea leaf, kind of like these, or the plant cells on the Elodea leaf, kind of like this picture down here. And then the solutions were either tap water, distilled water, or salt water. And so again, once you put that leaf in those cells in one of those solutions, now you have a concentration gradient, right? And then you're going to see some type of diffusion. So if we look at this, and then up on the top here we have animal cells. So what we see here are examples of each. So in isotonic, iso means the same. So if iso is the same, that means that there is going to be no net movement of water. We're still going to see some water coming in, some water coming out, just doing its thing, because again, osmosis is sort of always happening, but usually there's no net change. It's a net sort of neutral scenario. So in an isotonic solution, the concentrations are the same, am I recording? But there's no net change of water. In a hypotonic solution, we have, if I'm saying hypotonic, does the solution outside of the cell have more or less solute?
Outside of the cell?
Outside of the cell.
Less solute.
Less, that's right. So to say that again, a hypotonic solution has less solute outside of the cell. Because again, if we're talking about the solution, we're talking about the stuff outside of the cell. So with a it's fine. So hypotonic, we have less solute outside the cell. So if we have less solute outside the cell, where do we have more water?
Inside?
I hear both. So if we consider, you know, let's say you have, I'm gonna just show you an example. So yesterday, so if you had a cell, and let's say it had 50% salt, I'm just making this up.
Outside.
And then outside the cell had 10% salt. Is this a hyper or hypotonic solution? Hypo, right, because 10 is less than 50. And the solution, again, is what is outside of the cell. So if we use these percentages to show that they're either salt or water, if we have 10% salt, how much water would we have? Yeah, about 90%. I mean, this is arbitrary. So 90% water. And then inside the cell, if we had 50% salt, which would be quite a lot, we'd have 50% water, right? So now we can see, okay, does the solution or does the cell have more water? The solution, right? So in a hypotonic solution, we have less solute outside the cell, which means we have more water also outside the cell. The solute isn't going anywhere. The solute stays in the cell or outside the cell. So this is obviously not equilibrium, right? So in which direction would water move? Into or out of the cell? If you have more water outside the cell and less water inside the cell? Into the cell, right? Because osmosis is the diffusion of water. And diffusion means high concentration to low concentration. And if we're talking water, we're talking high or low concentration of water. These are kind of like big ideas to wrap your head around. But if we look at it this way, in a hypotonic solution, which is outside the cell, we have less solute. That's what hypotonic means, less solute. That means we also have more water outside the cell. So water will go inside of the cell. And we saw that with our Elodea cells, right, yesterday. Do you remember which solution was the tap, distilled, or salt, the hypotonic? Distilled, right.
Yeah.
So in your distilled cells, remember you saw the chloroplasts were kind of around the edge of the cell? And what's happening is that the distilled water was hypotonic to the cell, which means there's more stuff in the cell than there is outside. But there's more water outside. So water comes into the cell to try to find equilibrium. And the vacuole inside of the plant cell is where the water is stored. So the vacuole starts swelling with water, and that ends up pushing all those chloroplasts out around it, out toward the outside. And so that's kind of what you saw. So this is fine. With a plant cell, there's a cell wall which will prevent the cell from bursting. However, with an animal cell where there is no cell wall, if a cell takes in too much water, it can burst, and then it's dead, right? So that's obviously a big deal. Alternatively, with a hypertonic solution, we have, put those statistics up here too. So with a hypertonic solution, where do we have more solute, inside or out?
Inside.
Out, right. So with a hypotonic, so the solute is the stuff like salt or sugar or whatever. And hypotonic, we had less solute outside of the cell. Hypotonic means less solute. So hypertonic, we would have more solute outside the cell. Are we
So
sort of?
water's inside then?
Well, water is going to be on both sides. But yes, there would be more water inside. So more what am I doing? Hypertonic has more solute outside, and then we're gonna have more water than, we just said it, inside, right? Inside. So, if we have more water inside the cell than outside the cell, and it's a gradient, which way will water move? Out, right? Yeah, so water will leave the cell, and then hypo comes in. So, when we looked at our salt water then, so this plant cell at the bottom of the screen here, we saw that you had chloroplasts that were like shriveled up, not really shriveled up, but all gathered kind of in the center of the cell. And so what happened is that the cell was losing water, but it was losing water from that vacuole. And so we end up, and the cell itself too, the cell wall stays intact, but the cell membrane, everything kind of just shrinks. And so we saw the chloroplasts also kind of shrinking toward the inside of the cell. So again, that's dehydration of the cell. With an animal cell on the top, same thing. If we see water leaving cells, then those cells can shrivel up. So if we go back to our apple pie example, which I erased, but when you make an apple pie or a fruit pie or whatever, is that fruit, is that a hypertonic or hypotonic? Hypertonic, right? And I know that's what you want to think about when you're eating and preparing your fall pies. So this is hypertonic. This is so cool. So I know I'm spending a lot of time on this, but the reason why it matters is that homeostasis, right? If a cell gains too much water or a cell loses too much water, it can kill the cell, essentially. And so there are certain things we can do. You may have heard that, for example, salt can act as a preservative, right? If you think about like cured meats, for example. So if you add a bunch of salt to a solution, we see that it draws water out of cells, right? So if you think about that in terms of a bacterial cell that might be hanging around, or bacterial cells might be hanging around meat, and then we cure it. If you think about it as a preservative, it's going to draw water out of those bacterial cells, and then they're destroyed. So that's one of the ideas of sort of using salt as a preservative. That being said, some bacteria actually prefer salty environments, but aren't necessarily known to infect humans. So there's a lot more to say about that. But so I'm trying a new technique. We'll see if it works. Ask me 2 questions throughout the room that are not making sense about this situation. This is in lieu of me being like, do you guys understand? Do you have any questions? So give me 2 questions that you can think of that people are avoiding eye contact. That's okay.
Do you have like an easy way to remember hypo versus hyper, or how do you remember it?
Actually, I don't want to put you on the spot, but Charisse, were you the one that said hyper?
Yeah.
Do you want to share it, or do you want me to share it? Because I feel like that was a good, what's that?
You can share it.
So I'm going to put on, I already did, but Charisse had a great idea yesterday. With hypertonic solutions, we see that water leaves the cell, right? And the water leaves the cell because there's too much solute outside the cell, and so it's basically joining that. So the idea was, like and I'm probably not going to say it right, but when you're hyper, it's like you're going to leave, you're leaving. So, I don't know, I like that. Does that, maybe that helps.
Like hyper and gliding as well.
Yeah, you're hyper, you want to get out of here, just like water in a cell wants to leave in a hypertonic situation.
That's good.
In a hypotonic situation, hypo, it's not spelled the same way, but hypo, the cells gain a bunch of water and swell, like a hippo is a big animal, right? I don't know. So that's like funny mnemonic devices, Brent, that you had asked about. Like an actual scientific understanding is that, hypo gains water because there's less solute outside. But what were you going to say, Cameron?
What do you mean on the board when it says H2O out
Good question. So this is just sort of our statistics about these solutions. So in a hypertonic solution, we have more solute outside the cell. That's just what hypertonic means. So what that also means is that we have more water inside the cell. Or another way to look at that is you could say you have more solute outside of the cell, so you have less water outside of the cell. So because of that scenario, water is going to move out of the cell.
Okay.
And these are just like weird abbreviations, shorthand that I wrote up here. And then in a hypotonic situation, we have less solute outside of the cell, and we have more water outside of the cell. And so water will then move into the cell because there's more. Wherever there's more water, it's going to go the opposite direction. So if you have more water inside, water's going to come out. If you have more water outside, water's going to come in.
To compensate for the imbalance in the solute?
Exactly.
Okay.
That's exactly right. So that solute can't go anywhere. And if you think about, like, if you have salt water, and just in general, without talking about cell membranes, if you have a bucket of salt water and all this water evaporates, you still have the salt left over, right? So it becomes more concentrated as water leaves. So if you have, same thing with the cell, if the salt isn't gonna, if we look at this scenario that I put on the board here, if you have 10% salt on the outside and 50% salt on the inside, and water starts to go inside the cell, that's effectively going to increase the constant. It's going to go from like 10 to 20%, because if you have the same amount of salt particles, but you lose water, now your ratio of salt to water is changing, right?
Right.
Did that make it more confusing?
Yes.
Okay. Basically, the water moving will change the overall concentration of the whole solution by either diluting it or concentrating it. Yeah. So whether you want to use hippo or hyper or just like, I don't like the word memorize, but if that's what you have to do, that's fine. But in both cases, we're talking about water movement. And the extra credit opportunity I have for you will show this before your very own eyes in your kitchen or wherever. So I'll explain that.
Out there in the pie.
Not pie, but it's going to involve, actually, since we're on the topic, maybe I'll just tell you about it right now. And then we'll talk about active transport. When you, and I actually have this on Blackboard, I just have to make it available. Pause for a second. Can you guys remind me what type of energy does passive transport use and what type does it not use?
It uses kinetic energy, doesn't use ATP.
Right, so passive transport or passive energy is just movement from high to low concentration. We've demonstrated that with the iodine yesterday, with the glucose yesterday, with the perfume, and all those molecules just have kinetic energy, so they're just moving from high to low concentration. And then what is osmosis? Right, diffusion of water. So it's still diffusion, but it's the movement of water. And we will typically see that presented as either a hypertonic or hypotonic situation. So that's all fine and well, but we also have what's called active transport. In active transport, the particles are, for some reason, they can't move by diffusion, and or they're moving from an area of low concentration to an area of high concentration. So think about it, you know, you're a total rebel and you're going to run up the downstairs escalator, right? You're going to paddle upstream, right? These require more energy on your part. So when substances move from a low to high concentration, or if they move, we're going to look at a couple other examples in the cell where ATP is necessary. When ATP is needed, it is called active transport. One of the primary examples of this in a cell is called the sodium-potassium pump. And I have an animation up here, but it's kind of detailed. I'm just going to give you the quick ins and outs. Normally, in our cells, we have, or yeah, in nerve cells, for example, we would have more sodium outside and less sodium inside. Cool. You don't have to, I'm gonna use this to explain why it's active transport, but I'm not gonna ask you how the sodium potassium pump works on Monday, okay? But this is just an exercise. Normally then we have more potassium inside of our cell and less potassium outside. So if we just look at sodium, which is Na, if we normally have more sodium outside the cell than inside, which way would sodium diffuse?
Wait, can you say that again?
If we normally have more sodium outside of a cell than inside, which way would sodium diffuse? Into the cell, right. Diffusion is high to low concentration. Alternatively, if we normally have more potassium inside the cell and less potassium outside the cell, which way would potassium diffuse? Out, exactly. Cool, you guys are set up for A and P if you ever take it, right? So with the sodium potassium pump, so every time your neurons fire a nerve impulse, which they're doing tons of right now by just listening and looking at me, maybe you're hearing me but not listening, get it? Like, oh my God, I'm done. But the point is, after your nerves or your neurons go through this process and there's sodium coming in, there's potassium coming out, all the ions are in kind of disarray. And so the sodium potassium pump works to get it back to normal. What the sodium potassium pump does is pump sodium outside and it pumps potassium inside. Whoa. But we already have more sodium outside. Why are we pumping more outside? So if we're going from a low to high concentration now, that requires ATP. So again, this is an example, and the animation there, you're welcome to watch on your own, and it's fascinating, but it's not necessary for our purposes. But the point is that because we're pumping ions against their gradient, which means from a low to high concentration, we need energy to do that. So ATP can only be produced in living cells. So active transport can only happen in living cells. Was it my A&P class or you guys that I talked about rigor mortis? Yes. So rigor mortis is the concept that post-mortem, post-death, muscles are in a slightly contracted state. And one of the reasons, or the reason, is that, again, ATP can only be generated in living cells. And whenever your muscles contract and relax, to contract, it actually is breaking ATP, but then we need ATP to relax the muscle. So again, that's like a week and a half of A&P covered, done, right? But the point is, ATP can only be generated in living cells, so when the cells are no longer living, that ATP is not available and the muscles are still contracted because ATP is needed to break the contraction. Over time, I don't know, 3, 4, 6 hours later, maybe closer to 12, calcium diffuses out of the cell and that breaks the contraction. That's more info than you need to know, but I'm trying to make the point that ATP can only be generated in living cells. So, all right, cool, good times. You're laughing, just a lot of information, or yeah. Active transport, ATP. Hello. Hi. No, that's okay. We're just talking about rigor mortis, so it was a little bit. Oh my gosh.
Students aren't in that state, you're okay.
No, yeah, we're good. Thanks. There's a chair here. So the last part of, and actually I'm okay if we just get through these last couple examples here. The other type of active transport is through, I know you guys want to talk about vesicles one more time, right? Our good old vesicles in cells. So if you remember from Monday, when a vesicle makes a product that has to be secreted outside of the cell, the vesicle will be formed, come off the Golgi apparatus, and then travel to the cell membrane. So whenever something is being dumped outside of a cell, it's called exocytosis. Think exo was like exiting, okay? And so for example, you have sweat glands in your skin, and those glands produce sweat, and then will dump that sweat to the outside surface through exocytosis. Endocytosis, on the other hand, is bringing things into a cell. And that also will, basically the cell membrane will sort of hug the material that it needs and then bring it inside in a vesicle. So, and I'm actually not going to go through those other, there's some more specifics, like phagocytosis is cell eating, like particles. Penocytosis is more of like a cell drinking, like a liquid or a lipid droplet. But for our purposes, active transport either requires ATP to move things low to high, or is required to move big vesicles into and out of a cell. Do I have a picture? Oh, here it is. So this, I found, you guys know, there's Beatrice the biologist. It's like a meme generator. There's a Facebook page called like I linking love science. Can't say it.
Oh yeah.
You know what I'm talking about? But there's all these funny memes. So I found this one. Amoeba hugs are often fatal.
Follow it.
It's cool. So amoeba is a type of organism that will engulf through phagocytosis their food. Bacteria or white blood cells do the same thing to bacterial cells and kill them. So it's cute, isn't it?
So cute.
And actually, this. Do I have the picture of exocytosis? No, I don't. I like, these are just cool pictures of micrographs. Like, see vesicles, you guys. They're awesome.
I love this.
I'm just getting a vibe that we're on information overload, which is good because we're done with this. So,
Wait, can you go over what we need to know done for Monday?
yes, good question.
And
So,
the study questions.
for Monday, everything should be